Hi everyone, it's Tammy Shuro, and I have broken this week's content up into three separate narrated PowerPoints, kind of divided it into chunks that tend to build on one another. So this is number one, and it's really going to just focus on an introduction to quantitative research methods and specifically the randomized controlled trial. So our objectives is to know the meaning of a true experimental design in the quantitative paradigm and what that looks like. We're going to explore the three types of quant research, define what a true experiment is and what is required for something to be deemed a true experiment. We're going to be able to identify those when reading research literature. Sometimes there's a giveaway in the title, but not always. And our goal will be able to identify particular strengths and weaknesses of experimental design. And in our synchronous time together, we will look at a randomized controlled trial with our breakout rooms and whatnot. So how the course is kind of set up is that over these next three classes, you'll be learning about how research is conducted in this positivist paradigm, the quantitative method. And as I mentioned, we're going to look at three different main types: the true experiment, or the randomized controlled trial, or randomized design. We're going to look at something called quasi-experimental and non-experimental designs. And then we'll look at the quantitative appraisal framework and facilitated practice. And that will be the third class, where we'll actually appraise an article together. So by the end of that three classes, you'll have sufficient theory to begin the practice of quantitative appraisal. And I just want to say that again, in the third year block courses, they are all built around Health 2250 content, and that'll actually be assignment in one of the third year blocks is for you to appraise either a quant or qual article. And I'm always so impressed to see how students can move this content forward into the following year and look at research that's related to their specific vulnerable population they're working with in their blocks. So some of the strengths of quantitative design is certainly it gives you quantity, it gives you numbers and values and what have you. So that's quite different than qualitative. It provides objectivity, so it eliminates a lot of the subjectivity that could, impact a research design. It often is designed or utilized for predictability, so that we can look at cause and effect, and we can also, look for sure for cause and effect, but also there's correlations or relationships between the treatment, the intervention, and the outcome. So we aim for both of those things. It allows us to collect data through multiple methods. We might be collecting biophysiologic data. We might be using a tool to measure stress or depression or anxiety, quality of life. So observation, we might even include observation. And then it allows us to make comparisons between populations or groups. So we often have a control group and a experimental group. And then sometimes we'll do comparisons over time. So, some randomized control trials might be a one-time data collection, but some of them might be conducted over, months and months and maybe even a year. And then the benefit of the quantitative design is to be able to generalize the findings to the larger population. So we know we can't, sample the entire population. So we take a smaller sample of that, but ultimately If the study is tight and well designed, then we hope to be able to generalize those findings to that broader population. Definitely some of the weaknesses or the challenges are you have to know the topic in depth to be able to ask the right questions. And the challenge is often asking a question that can be answered, which I talked about last week. And typically in class, we do some practice with this. And I think students are often surprised how difficult it is to word a question that is answerable. Sometimes we get results and we think, well, what do those results mean? So the blood pressure drops by 3 millimeters of mercury based on that particular drug or treatment. Is that clinically significant? Sometimes, it can be an oversimplification of a very complex reality. Sometimes we run into challenges especially when we're using tools to measure more abstract concepts. Okay, say depression, for example. People are reading the questions, and how they interpret them might impact how they answer. Whatever we're studying has to be measurable. We have to be able to quantify it in some way, and some quantitative research can be fairly expensive. So you're getting to know that I love tables and graphs and charts and such. So I created this because I found it helps students understand the three kind of types of quantitative research. So we're going to start on the far right. So there's two types of quantitative research that actually test an intervention. So the researcher is providing some type of intervention, some type of treatment. Just remember, though, that an intervention or a treatment doesn't have to be necessarily, a drug or a procedure or those types of things. Interventions can be a support group. It could be a innovative teaching program. It could be I can't think of another example right now, but anyway, think broadly when you think intervention. Could be, okay, I've thought of one, could be like social support, testing the effectiveness of social support on maybe stress or depression or something in, patients who are on kidney dialysis. So on the far right is the experimental design. So this is the classic RCT, sometimes called the clinical trial. So the other one, the quasi-experimental, is sometimes called like a RCT wannabe. It usually has a number of the features of the randomized controlled trial, but lacks a couple of them. So we're still testing an intervention. On the left is something we call descriptive or correlational research. So it's still quantitative, but we're not testing an intervention. We're looking to describe something that's already occurring in that environment or that situation, and we're looking to see a relationship or a correlation between usually two things. So we simply describe that. We're not saying that when A happens, B happens, but we're just describing that situation. And the very, very simplest form of descriptive or correlational research is a questionnaire or a survey where we simply collect data, numbers, we quantify it. Example would be market research, when they survey people about what's their favorite cookie brand or their favorite car or whatever. So let's break it down a little further. That experimental design, or that RCT, you're going to see three essential features. There's going to be a randomized sample. So how they draw that sample is purely randomized. and we are going to look at sampling a little bit more next week and sampling strategies, but just know it's a randomized sample. There's going to be a control group. So you're going to have your experimental group and your control group. And the researcher is going to manipulate that independent variable, which is the treatment or the intervention. And as it says there, it could be, it could be a treatment, as in medication, new procedure, new treatment, but it also could be an education program, a support group, that type of thing. So, number 2, the quasi-experimental. So, you're still going to see manipulation of the independent variable. We're still testing that intervention, and the researcher is controlling that. Some people are getting it, some people aren't. And but the difference is that we do not have a randomized sample or a control group. We're missing one of those features. And what I'll say is quasi-experimental design is quite common in healthcare, because sometimes it's unethical to withhold a treatment from a group of individuals, especially if it's considered the standard of care. And so often we might lean toward quasi-experimental design. And I'm gonna, a couple of examples of that would be a cohort study. So I was involved in a study here in Calgary where we're looking at a community outreach program for preemie parents and babies. It's the only kind of that program in Canada, and we couldn't withdraw that program from families to see how did this group do compared to this group, because that was the standard of care. So we had to look across Canada for another group that didn't have that level of follow-up and care in the community post-discharge. And we found a group in Toronto. And we actually just finished our manuscript and are submitting that research study to a journal. So hopefully that's going to happen before the end of this course. And then lastly, the non-experimental design I've already alluded to, lacks manipulation of the independent variable. We're not testing an intervention, descriptive correlational question survey type of research. So there is a video loaded on the Blackboard site, and I really love that video, and I think it really makes these 3 types of quantitative research understandable. This is the little flow chart that she presents in the video, and I find this really helpful if you were given a scenario, a research abstract, and it didn't state exactly what type of quantitative research it was. You can use this little flow chart and you can say, is there an intervention? Okay, if there's an intervention, we know it's one of 2 things, right? RCT or quasi-experimental. So if there is an intervention, then we say, is there a control group? If there is, yes, perfect. Then we go on. If there wasn't, we say it's non-experimental. If it is, great. Was it a random sample? Boom, then we know it is a randomized control trial. If there isn't, it's a quasi-experimental. So it allows you to easily kind of discern by following the flow chart. So this would be something you would want to have handy when you write your midterm. And it is an open book exam, so you'll be able to have all these resources handy. So the RCT or experimental design, it's associated with the word science. As I mentioned, it's used to test cause and effect and relationships between variables. So the independent variable and the dependent variable. We're just going to look at that in a couple slides. So also known as the scientific method. So looking a little deeper at the randomized control trial, it's a study in which similar people are randomly allocated to 2 or more groups to test a specific treatment or intervention. So sometimes you might have actually 3 groups, and maybe one group is the standard care cohort, and the other 2 get different treatments, and we're comparing them all, or maybe one gets a placebo. And so it's the comparison of the standard treatment or control group to no treatment in that case. And of course that would have to pass ethical approval. So it is what we call the gold standard. It's the epitome of the true experiment. And certainly it's been held to high standards in healthcare. It helps us to really test interventions and to be able to improve on, patient-centered care, evidence-based practice, quality of care. So a little picture about kind of the flow of an experimental design. So it's a flow chart of participants, enrollment, and their randomization. And often at RCT would provide a little diagram like this. So it really helps to spell out exactly what's happening in the study, how they drew their sample, and how it evolved over the course of the study. So once again, 3 things. We've got the manipulation of the treatment or the intervention by the researcher. We've got a control or comparison group, and we've got a randomized sample. So you can ignore that last statement, see article flowchart. That was for a previous article I used, and we're using a different one this time. So here at the top, we have the assessment for eligibility. And this is really key in a randomized control trial. We call it inclusion and exclusion criteria. And so you have this pool of people that are eligible, and then for different reasons, some people are eliminated because they could impact the internal validity of the study. So there might be features about those individuals that might impact the results, and then we were looking and determining that it wasn't actually the treatment or the intervention that caused the change. So all the studies we look at in these next 3 classes will pay attention to that inclusion and exclusion criteria. Then those that meet the criteria are enrolled, and then they are randomized to the experimental arm or the control arm. And we're going to look at ways that participants are randomized. There's like 4 different ways that we can do that. And then we're going to, after the treatment, follow up for the ascertainment of the efficacy and safety of the outcomes. So just to review a little bit about independent and dependent variables in a randomized controlled trial. The independent variable, as you can see in the little picture on the right, it is what influences or causes a change in the dependent variable. So it's the cause, it's the effect, it's the intervention, it's the treatment, and it has an influence on the dependent variable. The dependent variable is acted on or produced by the independent variable. It's the outcome that we're interested in measuring, and it is what is caused by the independent variable. So if we were looking at the impact of a social support program on quality of life in patients with congestive heart failure, the independent variable is the treatment program, and the dependent variable would be a measurement of quality of life. Or it could be a new drug that we're going to test, and we want to see the influence of this new drug or medication on migraines, and we're maybe measuring incidence of migraines, severity of migraines, frequency of migraines, those types of things. So I have 2 slides here to allow you some practice in identifying independent variables. So what I would recommend is you pause the narrated PowerPoint here and work on identifying in each of these 5 studies which you think is the independent variable and which is the dependent variable. And then go on to the next slide where they're identified in red and green. So take the time to do this little bit of work, and then you can validate how accurate you are in the next slide. So in the first study, they looked at the effects of exercise training on depressive symptoms in patients with congestive heart failure. And so you can see the independent variable is the exercise training that's provided, and then we're somehow measuring depressive symptoms in these patients. Number 2, this was a really interesting study. They looked at the effectiveness of multimedia learning education programs on knowledge, anxiety, and pressure garment compliance in patients undergoing burns. So a lot of times if patients have a significant burn, especially a 3rd degree, they have to go through grafting, they often have to wear these pressure garments. And so you can see here where they had one independent variable, this multimedia learning education program, but then they measured 3 dependent variables. They measured knowledge, anxiety, and pressure garment compliance. This was another interesting stop smoking study. So they explored auricular acupressure, so that's the acupressure in relation to the ear, and they combined that with multimedia instruction or auricular acupressure alone. So here we've got 2 different independent variables. We've got the auricular acupressure alone, or we've got it combined with this multimedia instruction program, and they're measuring quitting smoking. And so they had some interesting ways that they measured quitting smoking, to the point where I think they collected saliva samples to see if they had some product that is produced in your saliva if you're smoking. Probably like survey, questionnaire, that type of thing as well. 4th one here. So this looked at growth, the length of parenteral nutrition, or what we call TPN, and morbidity, so complications or death or not death, that's mortality, sorry, morbidity, so any kind of complications this was in extremely preterm infants, and they compared a diet of exclusive human milk versus preterm formula diets. So here, the wording of it is such that don't get misled that what they present first is the independent variable and what they present second is the dependent. So in this case, the dependent, they're measuring growth, how long they were on TPN, and any complications, side effects, other illnesses that they had, in 2 groups, one receiving exclusive human milk and one receiving a preterm formula. One more. This was a comparison of 2 treatments on the length of stay of infants in hospital with bronchiolitis. And so they compared hypertonic saline and racemic epinephrine, 2 different treatments. And this was a really, really interesting study because it changed the course of treatment for how we manage bronchiolitis since then. So we used to always use racemic epinephrine, and that had a lot of side effects, and it had a real rebound effect when it wore off. It was expensive, and they actually found that the length of stay was shorter in babies treated with just the hypertonic saline. And so that's our treatment of choice now. So components of the true experiment, we've got researcher manipulation of the independent variables. The other main feature is control over competing or extraneous, or sometimes called confounding variables. And so there's two ways we go about this. We have random assignment or randomized sample, or sometimes called random allocation. It's called probability sampling, where we believe that by drawing a random sample, we are going to reduce the effects of these confounding variables that could impact our study results. And so we have some another way we try to control for these extraneous or confounding variables is that strict inclusion and exclusion criteria in our participants. So we're going to go on to look at the stress in nursing student research study when we get together in our synchronous, and we'll identify what some of the confounding variables might have been in that study. If we go back to the study where they tested the quit smoking program, you can think about different people coming into that study and their experience with smoking, how that could impact their success. Pardon me. And so, if you'd been a smoker for 20 years versus 10 years, if you had tried to quit smoking 10 times and failed every time, if you smoked a pack a day versus 5 cigarettes a day, like all those different features could impact on the results of the study. So we try and control those. Maybe we eliminate certain people from the study, and we randomly allocate in the literature review or the background, or sometimes labeled the introduction. And this is where we ask ourselves, did the researcher identify a gap in the literature supporting the justification for their study? If this is something that's been studied and studied and studied and studied again, how do you identify that it's worthy? Maybe you take a previous study and you add a new twist to it, and that's going to add to the knowledge base. The next thing we really look at in quantitative design is the research question. And this is where it kind of differs between qual and quant, where qual, they might just have a research purpose or aim or objective. They don't have a specific question articulated that they want to answer. But in quant research, we sort of expect that they would have a well-articulated research question. What do the researchers want to know? Where is this typically found in the research article? And typically it is often at the end of the introduction, the background, the lit review. Just before they move into the method or design, they'll usually clearly articulate their purpose and their research questions, and you might find this in the abstract of the article as well. So we're going to do some practice creating research questions when we come together. The research question should contain the variables and the intervention being tested. So, you're looking at the impact of kangaroo mother care on maternal depression, maternal stress, maternal confidence, etc. Okay, you've probably explored hypotheses and hypothesis testing in other courses, but research hypothesis really makes the question testable. And so we begin by looking at what are the independent and dependent variables of interest in the study. And then we look at how those variables have been defined, or what we call operationalized, so that they become measurable. So if we are looking at, we'll go back to the kangaroo mother care study. If we're looking at the independent variable of kangaroo mother care, how are we going to operationalize those things we're measuring, such as maternal confidence, maternal stress, and physiologic parameters in the baby: heart rate, respiratory rate, oxygen saturation. So we need to clearly articulate how we're going to measure those things. And so often we would use tools or instruments for that measurement. So, if you're using a weigh scale, or you're using a blood pressure cuff, or you're drawing a blood sample or a urine sample, those are biophysiologic measures and often more straightforward. The challenge comes when we're measuring these abstract concepts like stress, depression, anxiety, quality of life, etc. So the critical features of a hypothesis is it's the researcher's best guess as to the outcome of the study. I would hope in our RCT they would have a clearly articulated hypothesis, their prediction. So it points to the research designed to be used. It's going to suggest or predict relationships among variables, and it can identify the nature of the relationship. So there's kind of the two key types of hypotheses we'll look at would be directional and non-directional. So I'm going to give you an example and allude back to the kangaroo mother care study. So in a non-directional hypothesis, they would say there will be a difference in maternal confidence between mothers who are able to give kangaroo mother care and those who don't. So they're not predicting the direction, they're just saying there will be a difference. Then the directional would be where they would say mothers who are able to give kangaroo mother care have an increased confidence and decreased stress. So we're predicting the direction of the impact on the dependent variable. So I think this is one last slide here before we get into the next chunk on hypothesis testing. So the research hypothesis is what makes questions testable. So the question is not testable until a prediction is made, and predictions really equal the research hypothesis. And so two types of hypotheses are needed to conduct a test or a randomized controlled trial. The first one is there's a research hypothesis and a null hypothesis. So the research hypothesis, so we'll go on and we'll look at this in the Stress in Nursing student article. Is a research hypothesis included? And then how do we create a null hypothesis to be able to test this particular question? And we're going to dive more into both of those concepts. All right, so the null hypothesis. So if you think back to your stats class, there's no way to statistically model the research hypothesis. The prediction that there will be a difference, or it'll be directional, go either way. So it is necessary to define a null hypothesis. So a null hypothesis starts from the assumption of no difference or no effect. So null being no. And so we would say there will be no difference in maternal stress between mothers who do kangaroo mother care and those who don't. So we're stating there's no difference. The null hypothesis states there's no relationship between the variables being tested. No difference exists. Population is status quo. This is the hypothesis. This is the hypothesis that we actually test in a RCT or an experiment. So we're testing that hypothesis and looking to prove or disprove it. Okay, I think that